Okay, in the last video we talked about these things, cytotoxic T cells. Now we're going to talk about another kind of T cell, which is a T helper cell. Now they're similar. They both have the T cell receptor. They both got that. But in the case of the cytotoxic T cell, the T cell receptor is right next to this other protein called CD8. And in the T helper, it's a different protein, which is right up next to the T cell receptor called the CD4. Now, in the previous video, we saw that we looked at the cytotoxic T cell, and it recognizes the major histocompatibility complex 1. Okay, and that's expressed by all cells, and it will bind to that when it's got, remember, it's presenting an antigen. So that here's the major histocompatibility on the surface of the cell. It's got a little cleft in it, and it's usually got a little bit of your own proteins in there, just it gets ignored by the immune system. But if a cell gets infected with a virus, then you'll have a little bit of the virus. The virus has to get in the cell to do that. The virus is taken over the cell. You get a little bit of this is a little bit of the virus being presenting antigen in the this is MHC1. That will be recognized by the cytotoxic T cell, and the cytotoxic T cell will come along and start killing that infected cell. Now, we mentioned briefly, I think, that that can happen by itself, but it works better if you have T helper cells. Okay, now, T helper cells are similar to the cytotoxic T cells. The difference is they have the CD4 molecule instead of CD8, and this T cell receptor with the CD4, it doesn't recognize MHC1. It recognizes a slightly different molecule called MHC2, major histocompatibility 2. Now, major histocompatibility 2 is not expressed by all cell types, unlike the one. It's only expressed by B cells, which are another kind of lymphocyte involved in producing antibodies. We'll talk about them later. And it's expressed by these cells, macrophages, which, as the name suggests, they are phagocytic cells. Now, macrophages, so I've drawn one here. They exist all over your body. Right? They start off as a cell called a monocyte in the white blood. It's a white blood cell, and they migrate out of the blood, and they go everywhere, and they're just sitting there waiting to, if there's any immune problems to sort out. Okay, they're kind of fairly general sort of cell. Right, now what they do is they carry out phagocytosis. So what I've drawn here is a little bit of foreign material. Now that could be a bit of a bacteria, or it could be a virus, or it could be a virus with a few immunoglobulin molecules, antibody molecules stuck to it, whatever. But what the macrophage does, it comes along and it engulfs it. So it kind of grows a process out, and this grows out. Eventually it gets completely swallowed up, and it ends up in a vacuole inside the macrophage. There's a vacuole, and the macrophage secretes all kinds of all kinds of proteolytic enzymes and various other things, kills whatever it is and breaks it up, breaks the protein down into smaller bits. Now, when the protein is broken down up into smaller bits, so here is that bit of bacteria or virus or whatever it is that's been broken up, that will then migrate to the cell membrane, and it will be presented as an antigen in there. We've got a little bit of the peptide there, but this is not MHC1, this is MHC2, major histocompatibility 2, which can be recognized by, not by the T cytotoxic cell, but by this one here, the T helper cell. So the T helper cell, and of course the T helper cell has got the CD4 molecule in there, helping out the T cell receptor. This blue one is the T cell receptor. Okay. Just to remind ourselves that every different lymphocyte has got a different T cell receptor. How does that occur? Well, we remind ourselves, here we go up here. Right, this would be this cell here is in the embryo. How can one cell, you've got one gene for the T cell receptor, how can it give you millions of different T cell receptors? So what happens is the DNA gets spliced up, gets cut up and spliced together in different ways, so you get a slightly different gene in every single one of these cells here. So each one has a slightly different T cell receptor, so it recognizes a different presented antigen. Now these ones with the red crosses I'm drawing here, these are the ones that they recognize self. So in the embryo they are wiped out, aren't they, by apoptosis. So you end up with these ones, these recognize foreign antigens. The ones that don't, and they can be dealt with. And you may, there's as we said before, there's a T cell receptor for every possible combination of molecules you could ever get, half of which you will, well, 99% of which you will never see. But when anything new arises, there's always a T cell receptor that can bind it. Right, so there's the T cell receptor, and it's recognizing this bit of presented antigen. Now, what does the T helper cell do then? Right, T helper cells actually are subdivided into two types, so this is a bit confusing again, right? Right, now let's have a look here. So here's your activated CD4. This is your activated cell. It's bound to a bit of presented antigen. Right, if it's one type of helper cell, it will produce this substance called interferon, gamma interferon, that's a gamma. Or it might produce this IL-2, which stands for interleukin 2. Leukocytes are white blood cells, so interleukins are molecules that work between white blood cells. So these, both of these they're like kind of like local hormones. So they don't circulate around the body. They're hormones which act in a very, very localized region. So they're produced by one cell, and they will activate cells which are close to that. And what that will do is these gamma interferon and interleukin 2, they both stimulate cytotoxic T cells, which we met before. Cytotoxic T cells work okay by themselves, but they work better when you've got helper cells. It will make them divide more, and it will make them more cytotoxic. The other kind of T helper cell, these ones up here, type 2 ones, well, they produce different interleukins, interleukin 4 and 5, and these affect a different cells. These affect the B cells. They stimulate B cells, and we'll talk about B cells in the next video, but B cells are involved in producing antibodies. So that is the role of T helper cells. Very similar, well, they're similar to cytotoxic T cells. Differences are, of course, the main differences, we go back up here. Cytotoxic T cells, they have the CD8 molecule, and they recognize MHC1, and they deal with viruses which have been taken into the cell. The T helper cells, they have a CD4, they recognize MHC2, complex 2, and these deal with foreign material, not that has got into the cell and taken over the cell like the viruses, but it's just been taken in by phagocytosis there.